Adesso c'è anche la domanda. Va
ok,
bene.
ammettiamo i 213 dalla waiting room?
Mhm.
Sono tutti lì ammassati con la mascherina, spero.
Ok, ammettiamo i 213 dalla waiting room? Manolo, sento il mio eco per via di YouTube, credo.
Con la mascherina, spero.
Sì. Sì.
Manolo, sento il mio YouTube, credo.
Sì.
Ciao Angela.
Ciao Guido. Tutto bene?
Prendi appunti, mi raccomando.
Senti ancora eco?
Ci conto.
Ok.
Beh, magari sul corona, dai.
Li ammettiamo? Vado?
Vai.
Ditemi voi quando volete che condivida lo schermo. Perché questo Zoom è veramente potente,
Com'è? Qua. E join audio. Join video computer audio.
Io aspetto un segnale. Io aspetto un segnale per partire, ditemi voi.
Sì, stiamo aspettando che entrino tutti e poi partiamo.
Ok.
Buongiorno.
Posso mettere in condivisione?
No, aspetti un attimo.
Va bene.
Okay, now I think that we can start.
Okay.
Okay, there's no one left in the waiting room. We are more than 230 persons here, so welcome to all the participants to the first SIICA School of Immunology online. So I'm Silvia Piconese from the SIICA Junior Faculty. First, I will give the virtual stage to Angela Santoni, the president of SIICA, of the Italian Society for Immunology. And I invite Angela to spend a few words about the society for those of you who don't know it.
Okay, so welcome to everybody. I think this is a very challenging time for all of us, and we appreciated a lot your enthusiasm to adhere and participate to this webinar of the society. So the societies of immunology, clinical immunology and allergology has 600 members. I am the president. There is a vice president. At the moment is Marco Cassatella. And there is also a board of 10 other members. So, and 3 of them have expertise in clinical immunology. So the societies also have a junior faculty, which is very active. And among the different activities of the society, there is the promotion of educational activities in immunology, in clinical immunology and allergology. The major event is our National Congress. The National Congress was supposed to be in L'Aquila in September, the first week of September, and together with the National Congress wouldn't be there also the school, the SIICA school. Unfortunately, we had to postpone this. You can go ahead, Silvia, with the slides. So we have to postpone the meeting and the school. And so the junior faculty, together with Massimo Locati, decide to have this webinar on immunovirology. I should say this is the first webinar of the society, and we have been really, as I already said, really we appreciate a lot the participation, not only of Italian PhD students, but also from students of other countries, and I would like to welcome all of them. So I hope that you will enjoy this time that we are going to spend together, and I'm sure that there will be also prizes and so on. Okay, here is the SIICA Executive Committee. This was going to last until September 2020. Unfortunately, since we postponed the meeting, we are going to have we are going to stay also until the spring of 2021. There is one of our member of the executive committee, Professor Ennio Carbone, that passed away just 2 months ago, and this was really something very sorry for us, but we have now a new member that is Matteo Iannaccone that will join the board pretty soon. And if you give the next slide, Silvia. Silvia. Okay, this is the members of the junior faculty. And okay, if you go ahead, Silvia. And these are all the activities of the junior faculty, the organization of the school, but also the PhD retreat, the organization of the Univax Day, and this year again was very successful, and the organization of the International Day of Immunology, which is the 29th of April, and probably this year could be devoted to coronavirus and the immune response to coronavirus, and also a lot of exchange information protocol and so on. And all these activities, they are going, the next one, the next slide, Silvia. And also through all the social network. So please enjoy this webinar. And I leave the floor to Massimo Locati.
Okay, thank you, Angela.
Yes.
Manolo, if you can show the slides, I have just a few minor things to add. Okay, the previous one. This one is fine. Okay, so basically, this is just a very sum up of basically why we are here. So why we took the decision, and what I mean, I mean basically the junior faculty, to try this effort to set up a school. As Angela said, usually the society runs schools connected to the national meeting, which was supposed to be in September 2020, this time. We started doing schools in 2011, so it's a relatively established activity. But this one, of course, had to be postponed because of the COVID problem altogether with the annual meeting. So when this sad decision was taken, the junior faculty, myself, and a few other people in the society started thinking what could be the way, what we have to do with that, what could be the way to contribute from our side to the fight, to the virus, basically, and how we could actually keep the community alive and eventually increase it as it is this, let's say, a constant goal we have. And the idea was to try to have our immune system trained, as Silvia wrote in the flyer, improving their understanding as immunologists of this emergency. So the school has been moved from a general immunology school, as it is usually, to a focused set of webinars focusing viral immunology and vaccinology. And we, of course, because we are in a very unique moment, we have to experience new ways to do usual things. And as Angela said, this is our first trial with web-based, let's say, dissemination and teaching and seminars activities. So you will have to apologize upfront if something gets wrong. We hope not in a very dramatic way, at least. But we are also trying to turn limitations into advantages. This is another key message of this quarantine period. And we are now experiencing a moment where this school, which involves about 300 participants, over 200 PhD students as you are, 25% from abroad, coming, if you didn't miscalculate, from about 30 different countries. All these are absolutely uniquely possible because of what's happening. So I think there is already a learning message here, a taking home message, and that is try to do your best to turn limitations into an advantage, as we hope this experience will turn out to be at the end. Now, Manolo, if I can have the following one, this shows one of the two ways by which you can actually be involved in the school. Some of you did not fit in the class, basically. So they will have, in any case, the opportunity to follow the webinars as usual webinars through the YouTube SIICA channel. And this is the schedule, which is also available in the flyers which have been circulated and on the website of the society. However, for the most of you, the actual way to take advantage of the school is in the following slide, which shows you what the school itself is. And that is exactly the same program as you saw before in the pink sessions where webinars are advertised. But as you can see, in between one and the other, there are lots of activities ongoing. And these are all activities that, let's say, stick on the basic idea of what a school of immunology should be, and that is an interactive activity. Now, being interactive, sitting on your own couch at home is kind of a challenge. And for taking up of this challenge, I have to admit, as usual, the junior faculty did basically all the job. So you will have the opportunity, and I stress, this is not a must. I mean, it's an opportunity you can take if you really want, and I encourage you, of course, to do so, to work on groups, small groups, still inside the this device, this Zoom network we are in, to focus on dedicated questions which will be posed to you by the speakers, by the teachers. This work group will have independent management by the students themselves, but then they will be referred to the faculty and will be discussed, and you will have a feedback on that every time just before the beginning of the following seminar 2 days after that. These activities are 7 throughout the course, and will contribute to your participation in a kind of, if you like, game fashion, which will be ended up on the May 4th with a real championship, which I don't tell you anything about because otherwise there is no surprise on that. And at the end of the whole activity, there will be a score of your participation to that. And as Silvia will tell you much more in detail than myself, some prizes and awards to be awarded. Now, this is what we try to set up and to offer. And of course, no, that's fine, Manolo, you can go on. And of course, there are many people that needs to be acknowledged for that. You already saw, and I'm very proud to show you again the pictures of the junior faculty on the right-hand side, which are really key to this. Also, instrumentally is the SIICA secretary, Rita Repizzi, on the bottom, where you also get the email address for getting in touch with the society if you really want to become a new member, which we hope it will be for some of you at least at the end of this experience. But of course, the most of the thankful goes to the left-hand side of these slides, where you have, at least you can see once in the whole school, how the teachers look alike, starting from G.Poli-E and Elisa Vicenzi, who will be our teachers today, and Sarizza, Ennio De Gregorio, Johnny Scott, Stepanionic, Matteo Iannaconi, and Serita Cecchini. Program, actually, to be honest, but you have a program with yourself. So we really are very much grateful for them to spend some of their time with us for teaching us something that is not really usually on the main focus, at least for the Italian society. And I think this is something that we will have to work out a little bit in the future. So most of us, if not all of us, really are looking forward to improve our understanding and knowledge in this field. And that is basically all what I had to say, except for a in-house message, which is in the last slide. Okay, so be aware that all what will be shown to you basically is used exclusively for educational purpose. So everything you are exposed to, everything you can enjoy from this occasion should remain inside this room and should not be used outside for any purpose unless you really need it. And in this case, you first have to get a permission from the presenter. And this is something that we have to really take care of in a moment where following rules is important, as it should be basically all the time long. This is my contribution to the school. Having said that, I think I can leave the stage to the microphone, actually, to Guido, to start our first webinar on this SIICA of Immunology 2020 school. Thank you.
Thank you, Massimo. Before giving the stage to Professor, okay, we need a few more minutes to give the participants some rules on how to use Zoom. So, okay, for the Zoom users, please keep your video and microphone and sound off during the lecture. So you will see that sometimes the chairperson will mute you. But if you want to make a question during the lecture or after the lecture, we encourage you to open the chat, pay attention to write to everyone, write your name and surname if you registered with a nickname, and then write question. So in this way you will, the chairperson will know that you want to make a question, and there will be dedicated sessions to make your questions to the speaker. Obviously, before making your question, remember to unmute and possibly to switch on the video so we can see your face. And if you want to temporarily unmute yourself, just simply speak while keeping pressed your space bar. So you can unmute yourself by clicking on the microphone icon, or you can keep pressed the space bar. One last suggestion: at the end of the lecture, if you want to applause, you can simply click on the reactions in the bottom bar on your screen, or you can go to participants, click more, and there will be an icon for applause there. Next slide, please. So for the people who are following us on YouTube, if you want to make a question to the speaker, you can, and you can do it on Twitter. So you can post your question on Twitter. Please don't forget to tag us, and this is your Twitter account at SIICA, and please use a hashtag. And the hashtag is School of Immunology, SOI, SOI 2020. So in this way, at the end of the webinar, we will make your question to the speaker. And don't forget to become a follower of our YouTube channel. So for Zoom users, the instructions will be sent in the chat. So you can save the instructions on your computer and look at them also in the next days. Now I will leave the stage to Manolo Sambucci, who will introduce Professor G.Poli-E.Vicenzi.
Thank you, Silvia. I'm wanting to introduce our two speakers of the day. The first one is Professor G.Poli, which is full professor of pathology from SARS-CoV University, Milan. And I think most of you know him because he started his career in Mantovani Group and then joined the early 1925 lab in AH and Bethesda, and because maybe he's author of more than 250 publications, most of them of an HIV. The second speaker is Elisa Vicenzi, which is now actually leading as a group leader the viral pathogens and biosafety unit from SARS-CoV Hospital. She's also author of more than 120, including original articles, reviews, and book chapters. So, enjoying the lesson.
See, hi everybody. Can you listen to me? Can you hear me?
Yes.
Okay, let's try to share. Okay. Can you hear me?
Yes.
Now, yes.
Sorry, Apollo, but we have a lot of wind today, so I think this may cause some disturbances. Anyway, Manolo, eventually you just send me a message of some kind.
Okay.
So Welcome everybody. It's a pleasure to be with you, although at distances. And as we discuss how to construct this webinar, we thought about to do a two-part, to have a sort of a first a glance to a pandemic virus that we are much more familiar with compared to the new one, which is HIV. And as alluded by the introduction, thank you very much for the kind words. I've been working on this for 35 years, so it's been a really long journey. And in the second part, which is clearly much more cogent to nowadays, Lisa will take on, and Lisa, she's the real expert in coronaviruses and emerging viruses in general. So, okay, so let's see if I can go ahead now. Okay, so what I will do, obviously most of you have met HIV in your studies or perhaps by your research experience. And so a lot of the things that I will say will be mostly refreshers, but perhaps something will be anyway interesting of you. So we'll just start with a few historical notes. I will talk about very briefly about the origin of HIV, although I will not make a specific comment. I'm sure that Elisa will pick up and make some interesting analogies of what is happening nowadays with new coronaviruses emerging from 2002 onward. I will go briefly about what we call the natural history and the immunopathogenesis of HIV infection, perhaps the most clearly related semantic aspect to the SIICA Society of Immunology. A very brief mentioning of the state of the pandemics with a touch about Italy, and I will conclude very briefly with some current challenges. I will not mention specifically the issue of vaccine, although I will end up with that question about, but I will give you a clue during the presentation about the vaccine issue in HIV and by and large new emerging viruses in general. So let's start just with a few historical notes. Well, was more or less the same feeling that we are experiencing, or we had experiencing, at least in Italy, in the end of February. And we are back in June, the early June 1981. The less younger among us do remember well that period. We were just coming out of the 70s. It was a period of great freedom in general in the world. And something was happening as well in the field of disease in general. And there was a perception that the infectious diseases were not any longer a big problem for mankind. You know, we had a famous NIH director, for example, who discovered classified the cholesterol mediated diseases, that essentially thought that due to antibiotics and vaccines, the issue of infectious diseases were essentially well under control. There was a few moments of press in the world. It was the Legionnaire disease, toxic shock syndromes in the 70s, but you know, more or less they were essentially brought under control. So this was the general perception, the general climate that you know, society, including scientific and clinical societies, were living in this period. Then a very quick note, not really a scientific paper, but it was really a bulletin, which is called, as you can see, the MMWR, which is the Morbidity and Mortality Weekly Report that the Centers for Disease Control in Atlanta send again weekly and update the population, the American people mostly, on whatever is going on that is above, you know, the threshold of the noise, whether it is car accident or outbreak in nuclear powers, etc. That's still in the CDC, umbrella, so to speak. And as you can see in this very straight bulletin, it says in the period between 1980 and May 81, 5 young men, all active homosexuals, were treated for biopsy-confirmed pneumocystis carinii pneumonia at 3 different hospitals in LA. When they died, they have laboratory-confirmed CMV infection and candidal mucosa. And just as a note, now that the pneumocystis carinii is being renamed G.Poli-E-Vicenzi. So what strikes you is that these were young men, they were homosexual, they were people who essentially were healthy until that period, and that was a very severe and deadly disease that was coming out. And just to make the story short, and in a very rapid time for the technology that were available at that period, in 1983, the infectious agent was identified. It was identified, again, there was a lot of fuss about whether it was an American discovery, a French discovery, and the Nobel Prize committee decided that only the French should be acknowledged for the discovery. Well, essentially the Pasteur Institute was able to picture, as you can see on your right, a virus that was coming out from a lymph node culture of patients which were affected by this new disease. And notice in the title that we're saying patient at risk for the acquired immunodeficiency syndrome. This was the short acronym that was put forward because there was no clear identification of the disease at that moment. Yeah, it was called acquired immune deficiency to distinguish it from the congenital or primary immunodeficiency that were known at that time. And it was very clear at that point, particularly with a number of papers that Bob Gallo's lab published in 1984, that this was not only a new virus, but was a new class of viral pathogens that were exogenous, human exogenous retroviruses. And actually, there was a previous example. The very first member of this class of pathogen was not HIV, was a virus that was discovered in 1980 by the Gallo's group in collaboration with Japanese group. It was named HTLV1 for human T lymphotropic virus 1, or human T-cell leukemia lymphoma virus, as Bob Gallo likes to call it, which is the etiological agent of a relatively rare form of leukemia in adults, which is called ATL, or adult T-cell leukemia, and of a neurological disease that you can read about. So Gallo was sort of mistaken by the fact, he thought that the agent that caused AIDS was just a variant of HTLV1, whereas it was, yes, a retrovirus, but of a different family. And so it was very clear at the time, and this is still remind us what is happening something today, that after a normal, I would say, confusion, there was bacteria, there were fungi that were pop seeds that were, there was a lot of hypotheses initially about the potential causative agent of this new disease. But by the end of 1982, the scientists were convinced that this new deadly disease was caused by a blood-borne and a sexually transmitted entity, a virus. So basically 1 year after the discovery of the virus that caused AIDS, also the receptor for the virus was discovered. It was actually the CD4 molecule, because from the clinical and immunological side point, the most remarkable aspect of this new disease was the fact that these people had a severe, and sometimes almost complete depletion of the CD4 T cell subset of T cell, of lymphocytes, whereas the CD8 subset was unaffected in terms of number or even increased in terms of percentages, such as including also other type of subset. Then the physician learned that between the time of infection and the time of symptoms, an average of 8 to 10 years occurred. So it was a relatively slow disease, although deadly one. And it was very clear that this was not the whole story. It was just the tip of a gigantic iceberg. And note also the historical names that were given to the virus on the right on the slides. And again, this is analogy to what are we discussing nowadays, still waiting for a reliable serological test to understand how many people are actually infected with the new coronavirus. At the time, remember that at the time we didn't have PCR yet. So everything was based on serology, in that case. And again, Gallo set up the ELISA assay to detect blood and blood contamination, and therefore the safety of the blood was a big issue in those days. You might recall the issue about the hemophiliacs, which was another category of individuals that was severely affected by HIV/AIDS. So let me move briefly to the origin of HIV, that again, it's analogous to some extent to what we are experiencing nowadays. And the origin of HIV, again, is bring us back to the issue of our relationship with the environment around us and the perturbation of the environment. And essentially what we know nowadays is that there are HIV-like viruses that have been recirculating in primates for more than 40 million years. And during this very long time, at some point, there are factors that favor what we call nowadays the species jump. Okay, the fact that from one type of primate, these viruses can somewhat pass to the other one. And in some cases, particularly when the man perturbs the environment, we all have read, if you haven't read it yet, you can read Spillover by David Quammen, which is a great compendium that describes these events. And sometimes some of these viruses can affect humans. We have to be very, very clear about one thing, that because there are anecdotal episodes of infection from animals to humans, but however remains isolated events. It's only when a virus acquires the capacity to spread between human and human, that becomes a real major threat, like we are experiencing nowadays with the coronavirus and like what happened with HIV infection. And as already shown in the previous slide, but better highlighted in this one, is the fact that we had at least 4 independent cross-species transmission from the world of non-human primates, including one from gorilla, although the main transmitter, as you can see here, is the chimpanzee, the chimps. And out of this 4 type of viruses that have become human immunodeficiency viruses, the one that has become really dominant in terms of the pandemics is the M, so-called the group M of viruses. Then again, in 1986, a new virus came to the scene, it was called also similar to the previous one, but different enough to be named HIV-2. So the first virus was renamed HIV-1 or HIV type 1, and the new one was named HIV-2. Again, it was a contribution from the Pasteur Institute in this case, also in this case. And as you can see, the area, the geographical area that was affected was West Africa, is West Africa. And HIV-2 is similar but different than HIV-1. First of all, is very similar to its relative, which is the simian immunodeficiency virus. In some cases, they are almost indistinguishable. And overall, it causes a mild clinical course of HIV. And one of the main features of distinguishes from an epidemiological or clinical point of view is that it is very inefficient in terms of vertical transmission compared to HIV-1. And there are areas in which both HIV-1 and HIV-2 are present, and there are older studies and also newer studies that suggest that HIV-2 can interfere and slow down HIV-1 disease. Let me move now to the central part of my discussion today, which is the natural history and the immunopathogenesis of HIV infection. First of all, let me remark what are the routes of transmission of HIV-1 as well as HIV-2 to some extent. First of all, there are unprotected sexual acts. There is a gradient of likelihood of transmission from sexual acts. With, let's say, we are less likely and more likely modalities through which the HIV can be transmitted by via sex. But overall, sexual acts should be considered in R modality of HIV transmission. When there are, as mentioned, infected blood or blood products, and a huge problem that to some extent has been brought under control, which is the mother-to-child transmission. When we talk about mother-to-child transmission, it's important to keep in mind that in 3/4 of the cases, this happen either intrautero in the last trimester, because the infection occurs in the first trimester, which lead to abortion, or at delivery when there is a clear exchange of blood between the mother and the baby. But up to 1/4 of the cases are actually transmitted by breastfeeding. And whether it is quite simple, let's say in a context such as Europe or Italy, to tell a woman to stop breastfeeding his child or her child, sorry, you have to think about that in an African context, this could be really a very challenging thing because this sometimes is the only way that women have to feed their children. And what we have to say, although we will not discuss at length about this, this has been one of the most successful areas in which antiretroviral therapy, even with single antiretroviral agents, have been successful, because either one or two administration of antiretroviral therapy like AZT or DDI or other antiretroviral agent before delivery and after delivery basically shut down the likelihood of transmitting during peripartum period. If we look at the likelihood or the overall percentage of transmission, it clearly point out to the fact that sexual transmission is the dominant modality of infection worldwide. And just to remind you what is the UNAIDS position since 2009, please, all the young members and even the less young members of this webinar today, that the male latex condom is the single and most efficient available technology to reduce sexual transmission of HIV and of other sexually transmitted infection. And again, just to jump quickly on nowadays epidemic or pandemics, if you think about HIV, it's a fully preventable infection. We cannot say the same thing about coronavirus spreading because of the modality of spreading. But in principle, HIV could be totally prevented just behaviorally. Okay, sorry for the Italian slide, but I hope that also the non-Italian people connected today, we can understand. This is a very classical slide that it is very frequently used by my ex-former boss, Tony Fauci, but was actually firstly proposed by Tyndall and Cooper, David Cooper, great clinicians that died about 2 years ago in Australia. And it describes what happens in a person that is infected in the absence of any form of therapy. Well, basically, there is a first phase, which is called primary infection, that frequently is associated with an acute retroviral syndrome, but particularly in the early days was essentially misdiagnosed for mononucleosis-like type of syndrome. And then even in the absence of therapy, the symptoms go away and the person returns to his or her normal life. And that's the real trick that HIV does with humans, that apparently goes away. In the reality, it doesn't go away, it just takes several years before emerging with opportunistic infection or tumors. And in all this here, this is where the virus has all the modalities and all the opportunities to be transmitted by the modalities that we discussed before: sexual transmission, blood products, or mother to child. And only in the very final phase, we do enter in the so-called acquired immunodeficiency state phase, in which by 1 to 2 years, again, in the absence of therapy, in 95% or more of the cases, the person is brought to death. So this is where one of, taken from one review by François Barré-Sinoussi and others, and there are many, highlighting the main events in the HIV research. And again, 1984 is a very important year because not only thanks to the contribution by, mostly by Bob Gallo's group, the causative link between HIV infection and AIDS has been firmly established, but also because CD4 was identified as the primary receptor. But then we jump to almost 12 years later, we go to 1996 to have a very clear picture of how HIV can indeed infect its own target cells. And this because in 1996, with a sort of anticipation in December 1995, that we really understood how HIV infects the cell. Well, the anticipation is again a contribution by Bob Gallo's laboratory, and particularly by Italian group, mostly Paolo Lusso and Fiorenza Cocchi. So Italy has given a great contribution to this. But as it happened in science, they were pursuing a completely different question. They were looking for factors released by activated CD8 T cell that could suppress HIV transcription, a phenomenon that was described in the 80s by Jay Levy's group, that could again be used as a soluble factor to control viral replication. And they identified 3 molecules, but very few people around the world know at the time. They were 3 chemokines named RANTES, MIP1 alpha, and MIP1 beta, that they describe as suppressor factors produced by CD8 T cell. And in a few months later, then again, at Berger and many other groups around the world, describe that indeed CCR5, well, the first actually chemokine receptor that was engaged as a co-receptor for HIV entry was CXCR4. And then again, CCR5 was identified as a necessary entry receptor. So given to this contribution, now we know very clearly which are the milestones for which HIV can infect a cell. HIV can interact with his own GP120 envelope to the primary receptor CD4, inducing a conformational change which is absolutely required to interact with a second receptor or co-receptor, usually CCR5, which leads to the fusion of the viral membrane with the cell membrane operated through GP41 envelope, which is the second end component. The virus enters, and it's an RNA virus, but due to the reverse transcriptase, can convert its own genome from RNA into DNA and integrate to the host chromosome. And at this stage, the virus becomes a provirus. It's a very, very stable form, a permanent infection in these cells. And from this moment on, the virus or the provirus exploit the transcription and translation machinery of the cell to produce a new RNA and new proteins that will assemble into new viral particles that will be released to infect novel target cells. And but the receptor use of the understanding of how the virus infects the cell allow us to understand that the CCR5 is really the central element in explaining the pandemics. And in other words, if the virus could rely exclusively on CXCR4, it would have been much more limited in its capacity to spread. So the real goal is CCR5, and as you know, there are drugs such as Maraviroc that targets indeed CCR5. And again, when we learn a lot, we learn by, for example, that although 95% of the infected people would turn into progressors, that in the absence of antiretroviral therapy would end up with AIDS and die of AIDS in 8 to 10 years, there were a minority of individuals that were called defined long-term non-progressors. Our laboratory has studied them for a long time, that eventually may develop AIDS, but in some cases they can undergo for decades without any real sign of disease. And then there is an even less frequent type of individual that, although highly exposed to the infection, they do not become infected. And again, the importance of CCR5 comes here because a deletion of 32 base pair in the CCR5 gene essentially explains a great deal of both exposed seronegative and long-term non-progressors. When it is in homozygotic configuration, there is no CCR5 on the cell surface, so these individuals are not infected, or at least by the dominant CCR5 using viruses. When there is an heterozygotic configuration, they do have CCR5, but in a less frequent form, and they tend to become long-term non-progressors. And then there is an array of other mechanisms that has been associated to these rare phenotypes. So moving quickly to the main step of the immunopathogenesis of HIV infections. As we mentioned, R5 is the main virus that is transmitted, even from people that have CXCR4 viruses. So there is a positive pressure to allow R5 HIV transmission. There is acute clinical retroviral syndrome, which, as mentioned, looks like mononucleosis-like syndrome and was mistaken in the beginning. In terms of adaptive immune response, the first type of immune response that appears are CTLs, and antibody follows. And actually, CTL are indeed very important in the control of HIV infection, although they are not as important to lead to the elimination of the virus. So the central problem of the immune response in HIV is that, again, the CTL in particular, but we can expand this to include also other type of effector cells, including NK cells and other type of immune cell, it can control but never eliminate, never lead to HIV eradications. And when it comes to antibody, and this is clearly related to the issue of vaccine or developing a vaccine and the difficulty to obtain that, is that neutralizing antibody are indeed produced, but only late and at very low concentration, as the American used to say, too little and too late. So another very important question in HIV is what happened during the clinically latent phase. Where does the HIV live? This is a question that originally was provided and answered by Tony Fauci's group and Jeppe Pantaleo in particular, that you know very well, that he demonstrated that HIV basically hides in the secondary lymphoid organs. When you do an RNA in situ hybridization, you can see, removing the antibody component, that the follicular dendritic cell are really essentially a filter that retains a lot of HIV infectious HIV particles. And then 10 years later, another group at the NIH demonstrated that perhaps even a more important site are the MALT or the GALT, the GI-associated lymphoid tissue, which is really the primary target of HIV infection. And from that point on, it disseminates worldwide. But in addition to T cell, it's also very important to underscore that there are other cell types, particularly myeloid cells and microglia in the brain and tissue-resident macrophages that are infected through the same CD4, CCR5 dependent mechanism. And then there are astrocytes in the CNS that are probably the only example in which cells are infected through a yet unclear mechanism not involving CD4 or CCR5. And then just a brief touch on the great breakthrough that happened in the mid-90s, the advent of the so-called highly active antiretroviral therapy that now we refer to as combination antiretroviral therapy. And combination antiretroviral therapy take advantage of essentially the HIV life cycle of the virus that I referred to before, where you can target from the entry phase to the very last phase of the viral production. And by combining different type of cocktails, you can really achieve a very, very potent, very powerful antiretroviral strategies that are maintaining our people now alive. Now, somebody who is infected with HIV has a life expectancy almost identical to that of people of the same age with no infection. And antiretroviral therapy, combination antiretroviral therapy, has also changed completely the clinical fate of these people, because in the absence of antiretroviral therapy, as I mentioned, people were dying of either opportunistic infection or tumors. Now, essentially, the infected people die mostly of solid cancer, cardiovascular complication, and the complication of hepatitis C virus infection. So it's a totally new type of patient. Very briefly on the state of the pandemics, these slides from UNAIDS clearly connect the income of the person and the number of HIV infection. You can see the big outlier here is South Africa, and that has a relatively high income, at least in a part of the society. You can pick Italy on the right angle there, just to have a comparison. And that's the reason why HIV infection is now grouped together with malaria and tuberculosis with the so-called PRD, or the poverty-related diseases. Just a brief mentioning about the Italian situation. There are an estimate of between 120 and 150,000 infected people, although we are not sure that this is, actually we are sure that this is not the entire picture. And dramatically, about 20 to 25% of people nowadays in Italy discover to be HIV infected because they are diagnosed with AIDS. They go to the PS or to other clinical structures when we are already very sick, and only thanks to combination antiretroviral therapy, they can be reverted to a relatively healthy state, which means, and you understand that this is why the virus is still very highly present in our societies, because a lot of people do not know that they are infected and they keep spreading the virus as it happened 20 or 30 years ago. And, and again, just to conclude the link between therapy and the challenging problem, please remember that although as potent as it is, combination antiretroviral therapy controls the infection, prevents the clinical progression, but does not eliminate the infection. Which lead me to the very final part of this webinar. And back again to the mid-90s, when the powerful and the excitement to have combination drugs brought to the hope, or perhaps the illusion, that this could take care of the infection as well. The reasoning was quite simple, although it was elegantly put in an article in Science with full of mathematical algorithm. And simply the fact that there are multiple HIV reservoirs, that this virus integrates into the host genome, and there is a small pool of cell, an heterogeneous pool of cells, lymphocytes, but not only lymphocytes, that are not eliminated by antiretroviral therapy. And whenever, for whatever reason, you stop taking antiretroviral therapy, this small pool of cells can restart the firing, restart the infection in people. And so this is one of the current challenges. And there are a number of experimental approaches that so far have not been successful in terms of large available use. So there have been a lot of funding in this direction with very minimal results thus far, with the partial exception of the fact that at least in a few people that needed to have a bone marrow transplantation or transplantation of stem cell because of an occurring leukemia or cancer. And in the first case in 2009, this transplantation was carried on not only for HLA compatible donor, but also from a donor that was a CCR5 delta 32 homozygous. And this brought to the so-called the Berlin patient, Timothy Ray Brown. He defined himself as the Berlin patient. as the first person who was actually cured. And now, around 11 years after the transplantation, he has not taken any more antiretroviral therapy. And after this, last year in particular, there's been at least another one and probably another couple on the way, the so-called London patient, that basically recapitulated with a few interesting differences between the Berlin and the London patient, including the fact that he didn't have to have a total body irradiation, but a much milder type of pre-transplantation regimen that allow to have basically the same results. So for at least a subset of individuals, the cure is not just a hypothetical one. Whether this could be expanding to a larger population remains to be established. Well, let me finish this webinar, and before handing this to Elisa, with a sort of a summary and at the same time introductory note to the next webinar, which is a sort of a side-by-side essential comparison between HIV and the SARS-CoV-2 coronavirus. Well, for HIV, as we discussed, the virus is transmitted by sex and blood, and including the breastfeeding for mother to child. We know that there is about 37 to 38 million individuals that have been infected since 1981. We know that the virus has an intrinsic lethality of about 95% if in the absence of therapy, but we do have therapy. And this is really an important, a very important aspect of HIV compared to SARS coronavirus. We do indeed have combination antiretroviral therapy, but we do not have a vaccine. For the new virus, the new SARS coronavirus, as Elisa will certainly discuss in much greater detail, it is clear that it is transmitted by air droplets. There is still discussion whether aerosol can also be a means of transmission, and it is clear that it can be transmitted by contaminated surfaces and objects. The number of infected people is an estimate, and let's please remember that this all started just a few months ago. So if you look at the Johns Hopkins daily projection, we are in approximately 2 million of infected people, but we know that this is an underestimate between 5 and 10-fold. So we are rapidly achieving the numbers that HIV took almost 40 years to achieve. The lethality is also presumed because we do not know the actual number of infected people. And again, we are waiting for serological tests to tell us. But it is estimated also based on the previous SARS epidemics that will be between 1 and 10%. There is no therapy yet. There is a lot of attempts. There is a lot of concern also the usage of drugs on a compassionate base that, however, could withdraw the availability of drugs for other type of medical needs. And similar to HIV, the vaccine so far is not available. So the conclusion of my seminar will be the question for our students and difficulties and challenges for a preventative HIV and SARS vaccine. The first question, perhaps it's easier, which will be first. And the second question is less easy, or at least more tricky. Will they or it, depending whether there will be both or just one vaccine, confer a durable immunity? And I thank you for being with us, and obviously be happy to answer or try to answer to any question you have. Ciao.
Thank you, Professor Poli, for your interesting talk. So we have a question from our audience. Sorry, I'm Barbara Castani. I'm a member of the SIICA Junior faculty. So there is a question from Francesco Castagna. So Francesco, can you make directly your question to Professor Poli? So remember to unmute your platform. Francesco?
Yes, I am. I'm here.
You can go with your question.
Thank you, Professor Poli, for your seminar. And yeah, my question is just this. Which are the main reasons for the lack of a vaccine against HIV nowadays? I know it's a long question, it's a complex question and everything, but just briefly, if you can answer that.
Well, first of all, this is the question that you are supposed to answer. So I will give you just a few clues without giving you the answers. Suppose I don't think any expert in the world can give you a straight answer to that.
Okay.
Well, the first clue in my presentation, but in general, is that there is no existing example of spontaneous clearing of an infection, which is atypical compared to other viral or bacterial infection. In all viruses, in all viral infection, you do have a fraction of the people who are able to eliminate infection, thinking to the hepatitis viruses and many others. There is no such an example for HIV. The reason being that HIV is a retrovirus, okay? So that's the first clue. One thing that I should have mentioned, at least put a slide on, but thank you to your question, I can fill the gap, is that so far we have only one example of a partially successful vaccine. It again was 2009, a vaccine which was in a trial, in a so-called Thai trial, that provided a 30% protection for the first 3 years of infection. This has been attempted to be replicated in recent years. And just a few months ago, actually, we were already in the coronavirus epidemic, came the news that with the attempt of reproducing the results failed. But we have to say, and actually this will be part of your job to go in your group studies, there are substantial differences between the new, and I can provide you sidewise some of the key reference to look for it. There were substantial differences in terms of risk groups, composition of the experimental vaccine, readout population, that could have explained why the second experiment, so to speak, failed compared to the first one. So again, no example of clearing immunity in natural history of infection, and only one, let's call it weak or mild example of a potentially preventative vaccine with all the attempts that we have been doing thus far since 30 or more years of research.
Thank you very much for your reply.
I think there was another question, if I remember.
Yes, Prof. Hello.
Hi.
My name is Privilege. Yes, I had a question in line with issues that have to do with drug resistance, HIV drug resistance, especially in the African populations. I have noticed that there's a lot of work that is being done in terms of coming up with new drugs. Every now and then we have witnessed the launch of dolutegravir, which has come up with major advancements compared to the other drugs that have been produced before it. But I had a very simple question where I'm saying, will these drug interventions meet the resistance patterns that are being seen in these HIV infected patients?
You referring to HIV drugs, or you're talking more in general, including TB drugs, malaria, and other?
I can generalize that. Yeah, I'll generalize this.
Well, clearly, as you know, the Africa situation and the sub-Saharan Africa situation is a complex scenario.
Exactly.
There have been great improvement in the recent year compared to, let's say, approximately 10 years ago. But we are still not completed. There was, the so-called 90-90-90 goal that was supposed to be achieved. Exactly. Which means 90% of the people being tested, 90% of the people being put on therapy, and 90% of the people on therapy should follow the therapy. Recover. We are still back there, including there. Again, the issue that you, I think, alluding to is the issue of comorbidities and co-infection, which is a very important one, and the issue of drug-resistant strain.
Exactly, yeah.
And one thing I didn't mention, also because of the time, is that drug-resistant viruses can be transmitted, right? So if somebody and this drug-resistant virus emerge as a consequence of reverse transcriptase spontaneous mistakes, and of course, as a consequence of suboptimal therapy. This happened a lot in the relatively recent past, where you do suboptimal therapy simply because you don't have available drugs. So people are, maybe affected a bit in terms of clinical symptoms, but they do develop and expand drug-resistant strain, and they transmit the strain by, sexual blood, and this creates a new challenge for the medical community, because now your armamentarium of antiretroviral agent is greatly diminished. So it's a complex issue. It's a complex issue, and the answer, so to speak, is simply that there need to be implementation of programs where full regimen of combination antiretroviral therapy are brought in any area of the world, particularly in the area of the world which are most needed. And it has to do with drug price, black market of antiretroviral agents that you're probably familiar with.
Yes.
So it's a very relevant problem. And I think there are objective signs that the situation is improving, but we are not definitively conquered this aspect yet.
Okay. No, I understand. Thank you.
You're welcome.
Thank you very much. So there is room for other questions. So in the meantime, I would like to remind you to the people that are following us in the YouTube channel that you can make questions to Professor Poli, and you can use the Twitter platform to make it. So if there are no any other questions from our audience, we can move to the second part of our presentation from Professor Vicenzi.
Okay. Sorry, can you hear me?
Yes, we can.
Yeah.
Sorry, just a second.
Share the screen,
Yeah, but I have to get the presentation. Okay, oh my God.
Ce l'hai aperta?
Un attimo. No. Sorry. I need to open the presentation, but I don't know if I have to get out of the screen.
Sorry, we cannot hear you anymore.
Sì, adesso vediamo di attivarlo.
Okay. We can hear you, yeah.
Can you hear me?
Yeah, yes. Now we can hear you. Okay, perfect.
Okay, so Guido Macchietti. Okay, good. So good afternoon, everybody, and thank you for giving me this opportunity and talk to such distinguished immunologists and all the junior faculty and the PhD students. So my background is basically I'm a virologist, and I've been working for many years with viruses, and starting with HIV/AIDS. But in 2003, I stumbled in SARS coronavirus. And that was because I had the opportunity to isolate the virus from an Italian person who traveled to Vietnam and returned to Italy with atypical pneumonia. And just had the opportunity to have really a few hundred microliters of sputum. And so I obtained this sputum and tried to isolate the virus simply by putting the virus on viro cells. As you know, viral cells are very permissive cells to basically any virus infection because they are devoid of interferon response. So this is how a monolayer of viral cell looks like. And 24 hours after inoculation of the sputum, we obtain this phenotype. Basically, the monolayer lost integrity. You can see lots of dead cells, and basically a very cytopathic effect emerged very quickly after 24 hours of incubation, of cultivation. So what we did, we took the supernatant. So what first we did, we characterized by electron microscopy the presence of virions within the cells. And really cells were overloaded by viral particles, as you can see here, okay. And really, let me tell you, I have been working with HIV for many years, but I've never seen so many virions engulfing the cells. Really very, very productive infection. And just to show you a bit inset, okay, we have these vacuoles which have virions in it. And by negative staining, we could characterize the typical coronavirus shape with the spikes coming out of the envelope. As you can see here, this is a cartoon of the virus. Coronaviruses are RNA viruses, okay? The RNA is basically within this the nucleoprotein that sort of is around the RNA. Then we have the spike, the matrix protein, the envelope protein. Now, we did also immunofluorescence. We were quite fortunate actually to collaborate with Diasorin, which is a company based in Torino that's now developing lots of tests against coronaviruses, mainly PCR assay, antibody assays. But anyway, at that time, we developed an antibody. This was a murine monoclonal antibody against the M protein, which worked very, very well. And then what we did, okay, we sequenced the virus. Just on your left picture, you can see virions sticking out the membrane. So we took the supernatant, sequenced the virus, and found out that the virus was very similar to the strains that were isolated early in 2003 in China, consistently with the fact that this patient was traveling in China, in Vietnam, right at that time. So just, so basically what I did, I worked with this SARS coronavirus from 2003 to 2008. So I won't bother you with what I did in the lab, but anyway, I acquired the expertise now that I think will help me in working with the new coronavirus. And actually, we just basically obtained the virus, and we are setting up virus cultivation, virus replication assays. As you know, this virus is a typical BSL-free virus, which means that requires a containment which is quite stringent and limit a lot the studies on the virus. But nevertheless, I've started to work with the new virus. So basically, I want to show you data with the new virus. So basically, I will tell you the story of SARS coronavirus and then move on SARS-2 coronavirus, end up with some thoughts. So now let me go back to SARS 2002 and 2003. So this virus emerged in China, okay? And it emerged in the south part of China, in the Guangzhou part of China. And basically the virus went undetected for a number of months, I would say late 2002, beginning of 2003. But finally, it came up, it emerged really as an important pathogen in Hong Kong. And that was just because by chance, because a doctor who was the index case, who took care of patient in Guangzhou at the end of the 2002, he traveled to Hong Kong on February 21, 2003 to visit relatives. And the doctor stayed at the Metropole Hotel in Hong Kong. And he stayed at the 9th floor in room 911. And it turned out that after a few days, he went to emergency care and he died. And people who were staying in rooms adjacent to his room, and also people that stayed in rooms in front of his room, of room 911, got eventually infected. And from there, so we had the index case that infected 21 people. And then we had other secondary case with further transmission. And of course, there were cases without transmission. But anyway, to make a long story short, everything started from Hong Kong and moved to, sorry, I have a problem in moving the slides. So let me see. Can you hear me?
Yeah. Yes, we can hear you.
Sorry. So basically, in a short time, from Hong Kong, the pandemic spread to 3 different continents, including United States, Canada, Europe, Germany, Ireland, and Southeast Asia. And in a few months, well, I would say from November 2002 to July 2003, there were reported about 8,000 cases with a big toll of deaths, because the deaths reached almost 800. And the toll was paid by Canada, where there were reported 251 cases with 43 deaths. And I remember very well in 2003 when I went to meetings, and I remember very well the toll that Canada paid. There were many nurses that were put in isolation and in quarantine, and they were telling their stories that were quite difficult. And so thinking of what we are living now, I remember those stories, and I said, wow, when we first in January thought of SARS-CoV-2, I said, oh, I hope it won't come to Italy. And sure enough, I was wrong, and I was remembering really the experience of those nurses telling the stories. Anyway, so with SARS, we have been very lucky actually, because as you see, we only had 8,000 cases with a big toll of death, because the lethality was about 10%. And actually, next. Okay, and actually, I said we were quite fortunate, because by July 6, 2003, a Canadian newspaper, well, I was reported in a Canadian newspaper that WHO declared a victory over SARS, and Taiwan was the last country to be taken off list of infected areas. But as I said, with SARS, with SARS-1, SARS coronavirus, we have been very lucky. And the main reason is this one, okay? So with SARS-CoV, after an incubation time of 2 to 7 days, okay, severe pneumonia develops, eventually with acute respiratory distress syndrome that leads to death. And the pneumonia is quite long. You see, it can last from 20 to 40 days. And luckily, the virus shedding occurs during the symptoms phase, when the patient is basically in hospital or in intensive care units. So there is a lot of control of the situation, and healthcare workers of course, have DPI that they can protect themselves, they can protect the patient. So everything really was under control. And so given that situation where the virus is shed only during the symptomatic phase, so the isolation of confirmed cases and the quarantine of contact worked in halting the pandemic. And that was without vaccines or antiviral. And that's what we talk about these days. And so really from that experience, okay, that was 2003. And really then the virus was, disappeared. Okay, there was virus extinction. The main reason being the isolation and quarantine measure, because I remember also at the SARS time, we didn't have a vaccine, we didn't have antivirals, and they were not developed later on after the SARS finished. So, but the question was, a fundamental question was, could SARS come back? And sure enough, in 2012, we got MERS. Okay, so a new coronavirus emerged, and it emerged in Saudi Arabia. That was the Middle East respiratory syndrome. And so this virus is still spreading, so is still present with us. And we had got 2,400 cases with a big mortality rate of 34%. So this virus is quite dangerous. And indeed, what we know that this virus causes severe atypical pneumonia like SARS does. And differently from SARS, MERS has a prominent gastrointestinal symptoms and often renal failure. And and again, as I said, we have more than 2,500 cases, 858 deaths, and the majority of those cases clusters in Saudi Arabia. So, but then, of course, this is not the end, because now we are confronted with this new coronavirus. Just to give you a bit clear on the nomenclature, so the virus is called SARS coronavirus 2, where the disease that it causes is called COVID-19, which stands for Coronavirus Disease 2019. So just to give you a bit more information on this emergence, this is just a timeline of what happened at the end of 2019 and 2020 in China. As you know, this virus emerged in China, and it was already circulating by mid-November, and then circulating mostly undetected until the end of December. And on December 31st, okay, there was again clusters of atypical pneumonia that eventually exploded and diffused. And that was incredible, that by January 10th, virus was isolated and the Chinese published the sequence in PubMed. And this was a very important date, because having the sequence, it allowed to develop immediately diagnostics and possibility to develop vaccine candidates. So the sequence was made available to all over the world, and actually all over the world started to work on it. But again, with the hope that could never come to Europe, to the US and the rest of the world. And so the Chinese from January 10, they went on, And so clearly the epidemic grew more and more. And by this January 23rd, they decided to lock down the region, the Wuhan region where the virus emerged. And I'm sure you know this, panel because you have seen it many times. This is the Johns Hopkins University website that shows basically in real time what's going on all over the world. And this is actually the picture of China, where basically on their strict measure of isolation and quarantine, they halted the spread of SARS coronavirus to 83,000 cases with really 3,000 deaths or more. But eventually, so the question we had at that time, really, and we hoped very hard, we said, will China isolation and quarantine measures prevent SARS-CoV to become pandemic? And this, of course, wasn't right, because now we are confronted with such number. So we are almost like 1,800,000 infection. And this, like Guido said, this is certainly probably an underestimation. And actually, at least for the Italian numbers, we think that there are at least 10,000 more infected people that are affected by SARS coronavirus too. So anyway, with this epidemiological data that I believe most of you know, I would just to move a bit on more like the virology aspect and the coronaviruses aspect, just to highlight some of their origin and their ability, of great ability to adapt to their hosts. So coronaviruses are enveloped viruses. They are single-stranded positive sense RNA viruses. They have a huge genome because it is about 29,000 nucleotides in size, and is the largest RNA virus. And it is widely present in the animal world, including humans. And just to give you an idea, okay, this is a phylogenetic tree that is composed indeed by not all members of the coronavirus family. But what I would like to highlight your attention is this group here, okay, which is group 1b, which encompasses basically the coronavirus that cause the upper respiratory tract infection in adults. These are globally endemic and account for 10 to 30% of all basically winter common cold that affects every one of us. But so let's say that these are the good coronaviruses. Importantly, we have to highlight the bad coronaviruses, okay? The bad ones are those in the group 2b, and this one cause severe pneumonia, okay? So basically these ones infect the lower respiratory tract, mainly the lung, and they can cause really severe threat for humans that are infected. Now, another characteristic of coronaviruses is their zoonotic transmission. And invariably, all these pandemics that we had in the last 20 years, they are all been derived from animals. So they have a zoonotic transmission, meaning that they are transmitted from animals to humans. And certainly we know that for SARS coronavirus, and this wasn't clear immediately after the pandemic, so it took quite a few time to understand all the cycle of transmission. So clearly, okay, the virus originated in bats, in these horseshoe bats, was transmitted to civets, okay, which actually are the intermediate hosts, and basically they serve as an amplification host. And from civets, the virus was transmitted to humans, which actually appeared an accidental host. Just to remind you how bad are bats, okay? Bats are important reservoir of many coronavirus, but also other viruses, including, for example, the Ebola virus. And these are very special animals because they comprise 20% of all mammalian species, and with more than 1,100 species spanning 17 families. So they are hugely represented in the animal world. And just to give you a few information, these bats are basically long-lived animals. They can live up to 40 years. They are resistant to cancer, and they can survive infection, meaning that eventually the evolution and the arms race between virus and them as a favorite situation where bats are infected, but they don't get sick. And of course, the problem is that there are many opportunities of bat-animal contacts and therefore transmission, either direct or indirect to humans. Now, I alluded to MERS, okay, as I said, it emerged in 2012 in Saudi Arabia. And probably the reason for emerging in Saudi Arabia is clear from this cartoon, where again, bats are the reservoir of the virus, but the bats transmit to camelids. And as you know, in Saudi Arabia, there is a close contact between camelids and humans. But now what happens is, okay, there is a big difference between SARS-CoV and MERS, because eventually SARS-CoV was an accidental transmission from bats, as I said, from bats to civets, civets to men. That was occasion, okay, rare transmission. However, the transmission of MERS-CoV is continuous, okay? So there is a continuous transmission because of this close contact between camelids and humans. Now, if we move to SARS coronavirus 2, so we don't know exactly what's the chain of transmission. What we know is likely that horseshoe bats are the reservoir that could have transmitted directly to humans, and I'm going to show you in the next slide a phylogenetic trees that will convince you. And we don't know if we have an intermediate host that could eventually serve as amplifiers. And of course, this is very important to determine, because in the case of SARS, one reason that SARS was basically disappeared is because it was clear the intermediate host was identified. Because in that case, we had civets, and there was massive culling of civets in the Chinese markets. And that probably had to arrest the pandemic. Anyway, so we still have lots of missing points here. But just to convince you that really the origin of SARS-CoV-2, to look at this phylogenetic tree that was generated by this NextStrain website, where basically there are scientists that analyze in real time all the sequences that are basically published in PubMed or in another website. So basically they have the opportunity to analyze in real time and make phylogenetic trees and do comparison. And look here, how SARS-CoV-2 relates to SARS-CoV. Okay, certainly it is related. But what is striking is the relationship of SARS-CoV with bats viruses. And actually, I would like to point your attention to all these red dots and the blue dots. And the blue dots, is a virus that was isolated from a bat. in Yunnan, okay? And see, these viruses are very, very similar. They share more than 98% homology at the amino acid level. So we can fairly say that probably this virus is kind of, the bad virus is the ancestor of the actual SARS-CoV-2. Now, what's is interesting is this Wuhan bat coronavirus. And there is an important paper published in PLOS Pathogen in 2017 by Chinese group. And this group went to Yunnan, okay, in the region of Kunming, which actually quite far from Wuhan, because it's about, I guess, 1,500 kilometers apart. So they went to this cave because it's a cave where all shoe bats like to live. And basically they sampled bats for a period of 5 years. Okay, that was between 2010, 2015. So they went every autumn to sample feces, sample skin, sample basically bat material. And what they found is that these bats harbor, okay, SARS coronaviruses that basically they are indicated here. Actually, they isolated 15 viruses. They are here indicated in red. And basically, these viruses are separated, and they are basically importantly closely related to the SARS coronavirus 2. And these viruses not only are closely related to SARS coronavirus 2, but importantly, they use the human ACE2 receptor, which is the receptor that is used by SARS-CoV-1, but also by SARS-CoV-2. And so ACE2 is the angiotensin converting enzyme 2. Okay, this is a quite important enzyme, and because it converts angiotensin 1 into angiotensin 2, and it's an important regulator of blood pressure among other functions, and it's abundantly expressed on type 2 alveolar cells, the AT2 cells, and certainly it is AT2 cells are the target of SARS coronavirus infection. And ACE2 is the receptor also of SARS coronavirus 2. Here's just a cartoon of the structure. You see the human ACE2 and the receptor binding domain of SARS-CoV-1, but certainly now we have the crystal of SARS-CoV-2, and there's this receptor binding domain which engages human ACE2. There are different amino acids involved, but with a similar mechanism. So what happens is that what is scaring is that these bad coronaviruses, basically they use ACE2 receptors to infect human cells. As you can see here, this is the same paper, the PLOS pathogens papers. You see here on your left the bad coronaviruses, and you see the cells that either express ACE2 or don't. And in green, we have the receptor, and in red, the virus. And you see when you express the receptor, you have infection. And that's true for all the free coronaviruses tested, the bad coronavirus tested. And also you can see that there's a productive infection because if you use HeLa cells that express ACE2, you get productive infection for all these viruses. Now, so really what we should think of these bad coronaviruses, so we can make an analogy like, okay, think of an encyclopedia where you have many volumes and you have volumes 1 to 10. Volume 1 is SARS-CoV-1, Volume 10 is SARS-CoV-2, and among the 2 and 9 volumes, there are many bats coronaviruses that can be considered pre-pandemic, and these viruses, they use the ACE2 receptor, and they can infect human cells. And if they have the opportunity to jump, okay, they can be transmitted to humans and be very dangerous. So now I would like to focus on another important aspect of coronavirus, that is the great ability of coronavirus to adapt to their host. And so in general, coronaviruses have, they don't mutate very much. Of course, they are RNA virus, they mutate, but not as much as other viruses. And this is mainly because they have a polymerase with proofreading, so that corrects the mistakes that the polymerase makes during virus replication. But besides this, they have a great capacity to adapt to their host. And the important adaptation occurs in one critical point of adaptation is the receptor binding site of the spike protein, which is the protein, of course, that binds to the receptor. And you can see here, and this was studied, okay, this adaptation was studied, and there was identification of a precise amino acid in the spike protein. You can see on your left the two important amino acids that are the N479 and N487 in the receptor binding domain of the protein. Then after transmission to salivates, okay, there was a adaptation and changing in K and S, okay, the N479 changing in lysine, N487 in serine, and then jumping human, there was again another adaptation. So there's this all ongoing adaptation and selection. And to make a long story short, this adaptation have increased the binding affinity of the spike protein to ACE2. Now, so this is the bad news, but the good news is, this is the bad news, but this is also a good news, because basically with SARS-1, there was a kind of this adaptation led also to changes that by the end of the spreading infection, by the end of the circulation, so we had the early phase, intermediate phase, and late phase, and there was a progressive change in this receptor binding domain, which actually in the end led to a decrease of the binding affinity to ACE2. So it started bad, but it finished good. And again, another important mutation that occurred during SARS-CoV circulation in humans is this deletion, okay? Is the deletion of 29 nucleotide which occurred in ORF8. So ORF8 is here located here at the 3' end of the genome. It is a non-structural protein which function is still not known. We know which it works in the ER, and it's probably important in assembling the viral replication complex. And what happened is that, so the civets had a full ORF8 with the 29 nucleotide. The early isolates, late 2002, still maintain this 29 nucleotide. But then in middle 2003 and late 2003, there was this mutation, this deletion of 29 nucleotide that occurred in ORF8. Now, it took many years to understand what this mutation eventually did. And this was a very important work from Christian Drosten in Germany that was published in Scientific Reports in 2018 and took advantage of reverse genetics of SARS coronavirus. Basically, Christian Drosten could show that actually this deletion could attenuate viral replication in vitro. So you see here, so there are these, okay, you see here on the left with a wild type virus, and then he did other deletion, but look at the one on the right panel. So this is a plaque assay, basically, just for those of you that are not familiar with virus growth. So this is a plaque assay, which means that you take cells, like typically viral cells, you infect them with that virus, and basically you block the spreading infection by adding, say, a solid support like agar or carboxymethylcellulose or others. And basically you stop the spreading the infection, and finally you count the plaques, which means that are holes in the cells where the virus had replicated. So, but anyway, to coming back, so on your left, you see the wild type virus, whereas on the right, you see the deleted virus. So it's clearly that this deletion makes the virus, render the virus more virulent as compared to wild type. So just to sum up this part, so SARS-CoV virulence decreases with time during person-to-person transmission, and the decrease of virulence is explained by adaptive mutation in the receptor binding site of the spike protein and the ORF8. But now, so really we come back to SARS-CoV-2. And things that certainly you are all aware of. And really the danger of this virus is the community spread, because this virus is quite different from the SARS coronavirus 1, let's call it 1. So first of all, we all know that now, so the good news is that 80% of infected people basically are either asymptomatic or pouchy symptomatic. So they have a mild flu-like. But although they have a mild flu and asymptomatic, they can spread infection. And what is difficult is because these individuals don't know typically that they are infected, so they go out to the community and they spread the virus. So here there's important, public health measure that really persons that are sick, that have fever, that have mild symptoms, they have to stay home because they put in danger the community. Then we have that 20% of the transmission is severe, and here the danger is mostly for the healthcare workers, especially if they don't use the protective measure like the DPI and so on and so forth. So, so we are confronted with this important situation. And so I'm coming really now to the prevention and treatment strategies. And so I will touch upon on the vaccine first. Okay, so Guido's asked you a question, is an HIV-1 vaccine feasible? How difficult is compared to a SARS-CoV-2 vaccine? And of course, the answer is, well, I'm optimistic that the SARS-CoV vaccine will be eventually available. It won't be a short run, okay? But just to tell you that right now there are more than 50 candidates that are in studying, that people are studying, and the main target is the spike protein. Okay, the spike protein, and there are several strategies that uses the spike protein. We have mRNA vaccine, DNA vaccine, recombinant protein vaccine, vector vaccine, inactivated live attenuated vaccine. And just here on your right, you can see the time frame. Okay, an important thing is, of course, to develop in the right way and do not skip the phases that are necessary to have a safe and efficacious vaccine. So I'm moving quickly to the treatment strategy. So right now there are many trials ongoing. So there are more than 100 trials ongoing. And perhaps you may say too many, maybe, because there are several things that have been tested, and many trials perhaps don't have the power to give a definite answer. Nevertheless, so I would like to point out your attention to the antivirals, particularly this remdesivir, which is a broad spectrum antiviral, which is an adenosine analog, which is in phase 3 trial, and we hope that by April we know some results. And then we have this another nucleoside inhibitors that was developed by North Carolina University. And then, okay, just I found this cartoon that was just published today in JAMA that I found it very nice and sum up really the main strategies that are ongoing. Okay, so basically, so this is the representation of the virus life cycle. So from the step of viral entry, where we have the spike protein that interacts with the ACE2 receptor. But importantly here is to highlight the role of the function of this TMPR SS2 protein, which is a serine protease, which is necessary to activate the spike protein for binding and entry in the cells. And there is also already this compound that has been shown to inhibit viral entry, at least in vitro. Then if we move to the second step of membrane of the virus life cycle, like membrane fusion and endocytosis, we have this arbidol, which I guess you have heard a lot in the news. And then we have this chloroquine and hydroxychloroquine, which is kind of, you know, there's a lot of debate on usage of these drugs. And in my opinion, okay, they've been shown to inhibit viral entry and endocytosis. However, if you look carefully at the literature, I'm not sure that the data are quite convincing. Nevertheless, I think we need again to have clinical trials that prove their efficacy. And then to move on, okay, we have then RNA, translation, polypeptide. There was the use of actually HIV protease inhibitors. And it's clear now that these ones don't work. There is an interesting paper published in the New England Journal of Medicine that clearly shows no effect whatsoever of these HIV protease inhibitors. Then we move more to the RNA-dependent RNA polymerase. I mentioned the remdesivir. And then finally, nothing that we know about the late step of the virus life cycles that really is more involved in exocytosis and exit from the infected cells. So another important aspect which I think it is emerging and it's very interesting is the isolation of monoclonal antibodies that target the virus spike protein that prevents viral entry. And I recommend you a paper from Davide Corti and Antonio Lanzavecchia just published in bioRxiv, where they show that this antibody that's called S309, and actually this antibody was isolated from B cells of a SARS patient. Actually, this is the same patient from whom we isolated the virus in 2003. And so from this patient, they isolated SARS-CoV neutralizing antibodies. And they had the opportunity to have the blood drawn in 2013. And basically what they end up is with these antibodies, basically that is cross-neutralizing. So S309 neutralizes both SARS-CoV-1 and SARS-CoV-2. And they have identified the mechanism of neutralization, which is actually very interesting because it is independent of the receptor binding site. So these antibodies does not recognize the receptor binding site of the spike protein, but recognizes a glycosylate, another epitope, which is highly glycosylated, by the way. So I come up with lots of unanswered questions because this is a new virus. Actually, as I said at the beginning, I started to work with this virus, and it's really a very interesting virus, and from which we need to know more about its natural history, its origin, but really what's turning out very interesting is the viral pathogenesis, which is likely different from SARS-CoV-1, say. And of course, we need to know a lot on the role of innate and adaptive immunity, and we know basically nothing. And so importantly, the efficiency and timing of vaccine development, because this will be very, very important really to solve the problem of this pandemic, and a specific and versus broad spectrum antivirus. And I'm finished here, and I thank you for your attention.
Thank you, Lisa. Can you hear?
Yes.
Yes, hi. This is Mirela Cuca.
Oh,
hi, Elisa. Thank you for your talk. I will start with a question since I won. So my question is about your view on which are or which might be the main factor for the difference in spreading between SARS-CoV and SARS-CoV-2. Because you already touched some of the topics, maybe, I don't know, the idea that when you don't have symptoms, you can still spread the virus with this second SARS-CoV. This might be one reason. I just wanted to know, because we didn't have such a pandemic, right, with the SARS-CoV.
No, this was really quite I think there is a lot of the pathogenicity of the two viruses different. And as you know, really what is emerging is that all the problems of the coagulation system of this virus. So there is thromboembolism, and which seems very important. And actually, there is a lot of capillary problems. Okay, so we don't know if this virus infects endothelial cells or damages them or whatever. So I think the pathogenesis is different. So now, so just to say the pathogenesis, okay, so really one question is, okay, say if you get infected, okay, with symptoms, you have fever, so you are well for say 1 week, and then suddenly from say in a few hours, you may end up in an emergency room. So why do you have such quick change? Okay, if you're lucky, okay, after 1 week, you are healthy, you are back to normal almost. But why you can evolve so rapidly to really such a devastating disease? So this is unknown. Now, what I've seen, okay, this virus replicates very efficient in the upper respiratory tract. So whereas I don't remember such a feature with the SARS-CoV-1. So there is a tremendous amount of viral load in the nose, in the throat, which is obviously a problem because, of course, if you sneeze or cough, you have many opportunities to spread the virus. But so this is another important difference. And so it's really most what we don't know that we know. So that's why really it's important to study the pathogenesis of this virus. I don't know if I answered, Mirela, just
Yeah. Yes, for sure. I was also wondering about more of the severity, but also why does it spread so easily? If it spreads more easily than the SARS-CoV. You know what I mean?
Yeah, actually, if you look at there's this R0 number, this reproductive number, okay, which gives you an idea how efficient is transmission, especially at the beginning of the epidemic when you don't have drugs, vaccines, and so the numbers are quite similar, okay? It's not that it's very different. So basically the R0 is about from 2 to 3, so not much different. I think what could be different is that really this virus replicates efficiently in the upper respiratory tract. Okay, so there are more opportunities to spread it.
Okay. Thank you very much.
We have another question from the audience.
Yes, I have a question.
Yes.
Thank you for your presentation, and I just have a question. As you say, there is such a difference from SARS-CoV-2 and MERS. They are not same at in the genomic part, but just a question. Do you have any idea why the MERS was high mortality and morbidity in Saudi Arabia and all the Arabian countries, but the SARS-CoV-2 is not, they don't have a morbidity and mortality rate in that part? Is there any suggestion for a T-cell based vaccine if there is a memory there for that virus?
So you're saying that, okay, the reason why in Saudi Arabia the SARS-2 is not spreading is because they have memory?
Yes.
That could help?
Yes, yes. Or maybe there is any clue for this virus to find a good T-cell based vaccine, peptide based vaccine.
Well, I don't have an answer to your question. However, I was just struggled. So maybe, you know, when they tried to do the SARS-CoV-1 vaccine, there were quite a few problems in the animal models. That was mice and even monkeys. So there was a toxicity that was basically TH2 driven. So basically, when you immunize the mice, and then when you challenge, basically there is a TH2 response that is quite damaging the lung, okay, because there is eosinophil recruitment to the lung, a lot of IL-4 and IL-5 that damage the lung. So which is kind of concern, I mean, my opinion for really also the current SARS-CoV-2 vaccine, because this is something that should be taken in account. I hope it doesn't happen, but we shall not forget that it happened with SARS-1 in animal models. And SARS-1 vaccine was not moved, okay, mainly because, of course, there were no patients, because there was no possibility to test efficacy, because, you know, by July 2003, the cases disappeared. But also there were important toxicity in animal models that involved T-cell response.
Okay. Thank you for your answer.
Is possible to have a question?
Yes.
I'm a medical student from Trieste. My question is about the mechanism of spillover about the virus. So, which is exactly the molecular mechanism about the spillover? Because I don't understand how virus came out from bats or another animals. I think has smallpox or something like that. So can you give us more details about the mechanism, the molecular mechanism? So thank you.
No, well, not really. What we can say is just inferring, I mean, the sequencing analysis, an analogy between the bats viruses and the current human COVID-2. So really, it's just sequence comparison, okay? So, but I don't know how really, and just to say that they both use ACE2 receptor, and so, and I guess you require, of course, a contact, okay? And then you can become philosophical rather than mechanistically, okay? So it's easier to be philosophical, meaning that, of course, you can think of a One Health situation where you have the humans, you have the animals in the wild and the domestic animals. So we have these three worlds that if they come in contact, of course, there are opportunities for transmission. But really, this is more, again, I say philosophical than mechanistic. Indeed, I don't have an answer to your question.
Okay, thank you.
Okay, thank you. There is another question from Francesco Castagna.
Hello, thank you very much. And thank you for your talk. Just a quick question. As far as you know, is there any, among the candidates for vaccine against SARS-CoV-2, is there any fusion protein made obviously or of the one protein or more proteins of the coronavirus and other unrelated proteins or adjuvants? Thank you.
Sure. You know, probably there are, that I don't know, okay?
All right, okay.
I don't have enough knowledge to answer your question.
Okay, thank you.
Daniel Garzon. Okay, the other person is Eleonora Sala.
Okay, thank you for your talk. I would like just to know if it is known if interferon antagonists or inflammasome activators encoded by SARS-CoV-2 are different from the one of SARS-CoV-1, and this could explain maybe the different pathogenesis.
Yeah, it's a good question. I don't have an answer to that, sorry. Yes, but certainly something to think about.
Okay, thank you.
There is another question from N. Colomar. So the nickname.
Yes, sorry. First of all, thank you for your talk. And I just wonder if there is about this neutralizing antibody. Do you know if there is some clinical trial ongoing with this monoclonal antibody, or it's only been tested in vitro? Thank you.
Again, I don't know. What I've seen is just in vitro testing. I mean, yes, not even animal model testing.
Okay, thank you.
Okay, any other question? Anna Vanni.
Yes.
Yeah, Anna.
Yes, do you hear me?
Yeah, we can hear you.
I don't understand if the study about changing of protein amino acid of spike, amino acid of spike protein was studied on SARS-CoV-2 or only in coronavirus in laboratory. And if this sequence can be used for develop a new target drugs?
No, okay, for SARS-CoV-1, okay, the amino acid that I showed you occurred during virus circulation, so they occurred in people, okay?
Okay.
So then they were studied in a context of reverse genetic system in vitro, but that was mainly for ORF8. So to show their, difference, okay? So basically, they are real sequences, okay? They are real sequences that occurred in patients, in people, and but then proven to have an effect in the reverse genetic system. As you know, I mean, the beauty of viruses is that you can reconstruct them in vitro, okay? There are several strategies you can do it, but if you have this reverse genetic system, you can study mutation and you can study the effect of this mutation on virus virulence rather than several steps of the viral life cycle.
Okay, thank you.
Thank you. So we have a couple of questions. One from Mattia Ligotti. Actually, the question is for Professor Poli, if it is possible.
Well, I think, okay, I guess
Is still available. Otherwise, we can move to the other. I mean, there is another.
Okay, it's coming.
It's coming. Okay, thank you very much.
Okay, thank you.
But,
Hi, I have another teleconference in a few minutes, but I can.
Sorry, Mattia.
Very quickly. Sorry for that. I'm late.
No problem.
Because I have a problem with microphone. So just a quick question. For antiretroviral therapy, we know that the main goal of this therapy is the virologic suppression, but some patients respond in different manner, so with the viral blips or low level viremia. My question is, can genetic affect this kind of response?
Okay, let me try to give you a short but thorough answer. So let's say antiretroviral therapy nowadays is so powerful. So the drugs themselves and the combination of drugs have been so refined. And also related to your question, remember that in the beginning people had to take 30 pills a day, you know?
Yeah.
It was impossible. Now we are down to 1 to 2 pills a day, like aspirin, basically, or statins. So at least, let's say in Italy or in countries where antiretroviral therapy is covered by the state or by whatever public system, the people are actually doing well on drugs. We have problems, for example, in the United States or in other countries where depending on the kind of insurance that you have, the cost can be covered or not. Okay. This may lead to something that you alluded to, the fact that people can take suboptimal antiretroviral therapy, but certainly cannot control fully the replication of the virus, will lead to accumulation of drug-resistant mutations, and it complicates the problem. Okay?
Okay.
So there are two parts. So when it is optimally distributed and adhered to, I would say that the inter-individual variability plays a minor role. On the other hand, when conditions do not allow to have a full adherence to combination antiretroviral therapy, this may indeed be a problem.
Perfect. Okay. Thank you,
You're
Prof.
welcome.
Thank you.
Sorry, I have to leave. I have another point.
Okay.
Thank you very much. Thank you.
Ciao a tutti.
So, okay, yeah. So we have another question from Professor Vicenzi from Arianna Felicetta. Arianna, are you?
Yes.
Okay.
Can you hear me? Okay.
Yeah.
Okay. First of all, a really nice talk.
Thank you.
The question is, you mentioned a problem in the capillary fragility due to the coronavirus infection. Do you think this could be the main problem with the cardiovascular disease incidence?
Yeah, it could be. Yes, yes. I think so. I think, yes, sorry.
I mean, do you think that there is a correlation with the physiological role of ACE2?
Yes, sure. This is important question. Allora, so with ACE2, I think there is a very nice paper where in Cell, I can't remember the last name. Anyway, sorry. So anyway, but however, that was in the lung more than in the capillary. And so I'm going a bit out of your question, but I don't know if you wish to know the answer. Okay, no.
Yeah, sure.
Okay, so basically in the lung, there is the problem of angiotensin ACE2, okay, and ACE. ACE, which cleave angiotensin 2 into angiotensin 1. And when you have basically lung damage, okay, the angiotensin 1 binds to their receptor and causes lung damage. And here comes ACE2. Basically, ACE2 plays a protective role, okay, in this loop. And so what happens is that the virus, okay, binds to ACE2 and causes its downregulation, and therefore the damage to the lung is prevailing, okay? And so the idea is to give ACE2 analog, an ACE2 analog, first to block the virus, but also to prevent the lung damage by angiotensin 1 binding to their receptor. So this is the role in the lung, but I don't know what happens in the capillary or say in other organs.
Okay, thank you very much.
Sorry, the last two questions. One is from Simone Frascolla.
Hi, do you hear me?
Yes.
Yes.
Thank you for your talk. And my question is about the 29 nucleotide deletion of ORF8. I don't really understand how it does attenuate the virulence of SARS-CoV-2 virus.
Well, this deletion causes basically, removal of the open reading frame, so you don't have the ORF8 gene, okay?
Yes, this is clear, but
yes.
How does it act in a 20?
Okay, the function of ORF8 is not completely known, but is important to assemble the viral replication complex, okay?
Okay.
So if you don't have a virus replication complex correctly made, you lose infectivity, you lose viral replication.
Okay, thank you.
So the last one is from Eleonora Martinez.
Okay, can you hear me?
Yes.
Yes.
Okay, thank you for your talk. I was just wondering if there are rapid tests for the dosage of neutralizing antibodies.
Well, rapid test, what you can do is to do these pseudotype of viruses. Basically, they are viral-like, for example, viral-like particles, where you take, say, a lentiviral vector, which you pseudotype with the spike protein of the coronavirus. And you can, incubate these viral-like particles with the virus, and then infect cells and see if you see a reduction, a dose response activity. Of course, okay, you incubate the virus with your antibody, and then you infect these the cells. Okay, so it's basically mainly on pseudotype viruses, lentiviral based. That's what I've seen people using mostly, and there are several different versions. Okay. But then there is also like a VSV virus that can be used, and maybe herd is even more good and efficient, is better than the pseudotype lentiviral based VLPs. So with this assays, you can work in a BSL-2, okay? Otherwise, you need a BSL-3 if you use full virus, competent virus.
Okay, thank you.
No more questions from the audience, means that we can move on. Thank you.
Sorry, I have a question. I wrote it. I don't know if I can.
Yes.
Okay, thanks to Professor Vicenzi. I've been present to your presentation in Humanitas a little bit earlier than the problem was also here in Italy. And my question is how is it possible, not possible, but what's the role of immune system, because we have seen in many cases that patients with immunocompromised system respond better because takes drugs like Plaquenil. Can you give and explain about this?
Sorry, I cannot hear.
Hello?
Yes, I lost you.
Okay, no, my question was about the immunocompromised patients.
Yes.
They take drugs like Plaquenil.
Yes.
And when they take these kind of drugs, are protected, or the response is better against the virus?
I don't think there are data to answer your question, really.
Because in many newspaper, there is this, not Science Journal, but other newspapers, there is this this point about the immune system and the virus, and in particular about interleukin or others.
Yeah, sure. Yeah, there is this antibody against IL-6, right? All these drugs. But again, I think really what we need powered clinical trials. Okay, to answer all these questions about the drugs, and to do many trials and just do, say, unpowered trials, I don't know how much useful is. So, but anyway, there are many ongoing, and I think also trials are important. To say, okay, something doesn't work. That was clear with the HIV protease inhibitors, where there's no effect. Okay, this was very clear. So say you do the trial and it doesn't work. Okay, forget it. So, but important is to do it to do powered trial. Okay, with good statistical endpoints.
Thank you.
Okay. Thank you to the speaker, to the audience for the question. We can move on. And now, my colleague Barbara, she explained you this this couple of slides. Please, Barbara.
Yeah. Thank you, Manolo. So our webinar basically is over. So we would like, first of all, as Madonna said, to thank you very much our speaker for the grateful and very interesting talk. And we would like also to thank our followers in the SIICA YouTube channel. So please stay updated with our social network and for the upcoming webinar of our school. And